Hello, everyone, and welcome to this new Gen webinar entitled For Cell and Gene Therapy Products, Early Characterization is Critical. Today's webinar was made possible through sponsorship from Halo Labs. As usual, I'll be your host for today's event. I'm Jeff Bogaliskis, Technical Editor for Genetic Engineering and Biotechnology News, which is celebrating its 40th year, bringing you the latest information on the most cutting-edge tools and technology that life scientists need to keep their research moving ahead. So cell and gene therapies represent some of the most advanced treatment strategies in the clinical arsenal. And to create these cutting-edge medicines, biomanufacturers have developed equally advanced processes to ensure maximum efficacy and safety. As such, companies are always on the lookout for innovative methods to identify contamination, such as subvisible particles, which is the leading cause of drug product recalls. But the difficulty for many cell and gene therapies that are currently on the market is that the drug itself is subvisible in nature. So, how do manufacturers tell a good particle, like a cell, that should go into a patient from a harmful one? Well, in a few moments, our guest presenter is going to address that very topic. But before that presentation starts, let's first meet our presenter. Dr. Bernardo Cordovez is the Chief Scientific Officer and founder of Halo Labs, where he currently leads application development. Bernardo's background is in mechanical engineering, and he has extensive experience in particle analysis, detection and manipulation, drug delivery devices, microfluidics, nanophotonics, and bioenergy production. But today, Bernardo is going to tell us about some case studies measuring stability and purity of CAR T-cell therapies and AAV gene therapy products using his company's RCL instrument. Immediately following Bernardo's presentation, we're going to have our Q&A session. So I'm going to remind the audience to submit questions for Bernardo so that we can have a really good, lively discussion. We'll try to get through as many of your questions as you send in. To do that, it's really simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. I encourage everyone to submit as many questions as they can. But first, let's pass the mic over to Bernardo and hear more about subvisible particle identification.
Hi, thanks for having me, and welcome everybody. My name is Bernardo Cordovez, and today I'm very excited to talk about the RCL and how it can help you better characterize your gene and cell therapy products from the earliest of stages using low volumes. The aura is all about aggregates and particle impurities. At the basis of this technology, it images every particle present in your bioformulation, it sizes them, it images them, and it identifies them so that you know if the particles that you're seeing are actually your drug product aggregating or if it's an external impurity. One of the things that was very surprising to me as an inventor is just how ubiquitous subvisible particles in larger aggregates are found across the bioformulation spectrum. So in these image on the left, these are protein aggregates that are about 100 micrometers in size in equivalent circular diameter. And when you think about the fact that a protein is only, say, 5 nanometers in equivalent circular diameter, it takes a lot of instability to generate these particles, yet they're there all the time. As this technology has progressed and the field has progressed, now we're doing a lot of work in the cell and gene therapy space. And what we're seeing is that the AAV therapies are where the protein formulation was about 10-15 years ago. And these AAVs can be quite unstable and form large, subvisible, and even larger aggregates as are seen here. And in the case of cell therapies, well, these are potentially even a bit more challenging because cells can be easily above 1 micrometer in size, and they themselves can aggregate, potentially indicating a product flaw. Now, the nature of these particles can be inherent, that is the drug product aggregating as we're seeing here, or they can be extrinsic material. For example, fibers or other process contaminants that make it into these formulations, and you very quickly want to know if they're there, just in case they present an immunogenic threat. Now, subvisible particles are a critical quality attribute. And you'll hear me say the word subvisible particles quite a bit here. These are particles that are bigger than 1 micrometer in size, so they're large, but they're actually the #1 cause for product recall. They present a safety concern from immunogenicity, undesired immune reactions, all the way down to capillary occlusions, because the thinnest human capillaries can be smaller than 10 microns in diameter, so particles that are bigger than this can actually obstruct and produce anaphylactic shock. But more commonly, they've been tied to reduced drug efficacy. Perfectly good drugs that are aggregated, your body will not like those and produce antibodies that attack them. And more commonly, the presence of large aggregates of AAV or protein are an indicator of low shelf life stability. Because of all of these undesired effects, the FDA regulates subvisible particles, and there's very well established USP guidelines for the tolerable amount of particles that are larger than 10 microns that are acceptable. Now, typically, people think about particle analysis often at the very latest of stages, past formulation development, but what's critically enabling about this technology is that we can conduct subvisible particle analysis and understand every single particle across all of our samples from the earliest of stages as well. Now, let's talk about size ranges. How big of particles are we talking about here? Many of you may know about different technologies to size particles, for example, SEC to measure dimers or trimers of protein aggregates, AUC, dynamic light scattering. The particles that this system characterizes are larger than 1 micrometer in size, all the way up to the visible. So we're not looking at things as small as individual viruses, we're looking at aggregates larger than 1 micrometer in size. Now, why is that important? So let's assume that you were looking at this image on the left and you had a biologic sample, you put it in SEC, you conduct a lot of different stress tests, for example, shaking, and you see that the SEC signal doesn't change. So you could potentially assume, hey, I don't think I have a stability or particle stability problem in my sample. However, if you measure this exact same sample and characterize its micrometer scale, its subvisible particle content, you might actually see that those counts are through the roof. So just because you don't see small particles, it doesn't mean at all that you don't have large ones. It is not a continuum, and the dynamics of each size range is independent. Now, how does this technology count and size particles? It's actually quite simple, and it enables low volume, high throughput, subvisible particle analysis in 1 minute per sample. We start with our cartridge, our consumable plates. And these consumable plates, as you can see, they have a metal frame and a stretched polycarbonate membrane between this frame. And there are these dark rings. These are hydrophobic rings that enabled 96 independent test sites to be measured independently. Now, the way this works is that first you take this plate and put it inside the instrument. It characterizes the background texture of this plate. You then load and filter your sample, and then you measure it. It's that easy. Let me walk this in a little bit more detail. So as I mentioned earlier, first you take the plate, put it in the system, and the system focuses and images on every well that you selected. This is before sample has been inserted, and you can see there's a background texture, potentially some lines from the membrane roll. Maybe you might be able to start to see some of the pores. But you don't really want to mistake any of these features, of these textures of the membrane with the particles of interest. And that's why we characterize this background before we do anything. Next step is that we take these previously backgrounded plate and we put it in a proprietary manifold and we pipette, say 40 microliters or more of volume per well. We then vacuum the plate and think of it as essentially making lemonade with a sieve. The pores are 400 nanometers in size, so essentially particles larger than 400 nanometers will stay behind. And in the system, we're basically measuring particles larger than 1 micrometer in size. We then dry the plate from the bottom so that we measure dry filtrate following the USP standard protocol. And we reinsert this plate and image it again. And here's what you're going to get. Now, you might see these large particles, these subvisible particles, in this case corresponding to protein aggregates, but I still have that background texture. Now, this is something that you wouldn't actually see in the instrument because we have this original background membrane image. The system automatically is designed to align both of those images and get rid of the background texture on a pixel by pixel basis. So particles that were previously invisible will become visible. And you can resolve the smallest particles in high contrast. Now, high contrast is incredibly important because other techniques that measure these particles in a liquid, because the particles have the same optical properties as the surrounding liquid, you will missize them and undercount them. But that's not a problem here. Now, what are the different tiers of information that you get from the system? First up, you get images for every single particle. So not only do we get images for every single particle, but the information, morphological and dimensional information for every single one of them. In this case, this particle happens to have a 57 micron equivalent circular diameter. That means if you wanted to turn this protein aggregate into a circle, it would be 57. Its maximum extension is about 82 microns, and all sorts of other morphological features, including its aspect ratio and its circularity. We also have other light sources that allow us to understand how much these particles protrude out of plane, essentially like a 3D filter, with a proprietary light source called SIMI. So again, we've imaged every particle, and we have the sizing information from every particle. Now, let's look at an individual Welleb sample. This is one of the most popular graphs in particle analysis, and that is a particle size distribution. This tells you, it's a histogram that tells you how many small particles are present in a given sample and how many large ones. For biological samples, typically you'll see something that follows this trend. It's an exponential decay. There's lots of small particles and you really want to have very few big ones. Again, because those can potentially obstruct your capillaries. Now, we obtain this for every sample in a single minute. With comparable techniques that you have to wash the consumables frequently, that's certainly north of 15 minutes per sample. So we have every single particle size distribution for every well, but this is a high throughput system. So the Aura software enables you to basically name the different wells under different conditions, and it will intelligently group all of the relevant data. So for example, if I just give all of my wells in which I processed aggregated IgG, it will group all of this data, and we can see that we have 70,000 particles per mil greater than 2 microns for IgG and a very low CB, under 3%. We used a plastic that mimics protein aggregates called ethylene tetrafluoroethylene made by NIST. And in this case, that plastic that looks like subvisible protein aggregates measured 40,000 per mil, while the negative controls held. So we went from single particle to particle distribution to entire particle characterization across all of an experiment, 1 minute per sample, 96 samples in 96 minutes. So what are some of the things that people are doing, whether it's in pre-formulation and formulation development? Now what you can access is, for example, doing screening, particle screening for different molecule types under different buffer conditions. Here we see that we have 3 different molecule types: BSA, lysozyme, and IgG, and they all have aggregates, and they were formulated in different buffer conditions. And this chart is repointing all of the counts greater than 2 micrometers per mil. So the BSA was quite stable, and it didn't form many aggregates. It was most unstable at this B3 citrate buffer condition. The human IgG was also fairly impervious in this particular case for the different citrate buffer conditions, but maybe a little bit less under B10. Meanwhile, lysozyme, not exactly a therapeutic molecule, formed lots of aggregates, but it was actually the B3 that produced the least particles. So different molecules respond differently to different buffers, and the good news is that now we can do these high throughput subvisible particle screens in minutes. In this case, 96 samples in 96 minutes. If you wanted to do this with fluid imaging techniques, this is an experiment that would simply just take months. Now, this tool, one of the main advantages is that it is low volume. You can get quantitative data down to 5 microliters per sample. If you compare that to against competitive techniques, light obscuration takes milliliters, flow imaging takes hundreds of microliters, and using complicated techniques like just flowing in fluidic plugs and kind of a nightmare. But anyway, what this data shows here is that you can obtain linear data down to very low volumes. This tool itself doesn't solve statistics, right? So if you have very little particles there, it will report no particles. So as long as your samples are well mixed, the data will be quantitative, down to the lowest of volumes. In addition, it enables high counts linearity before saturation. And I'll be honest with you, to me that wasn't intuitive at the very beginning. But it's actually kind of amazing that in a 5.6 millimeter diameter well, you can fit lots of micrometer scale particles before they start stacking next to each other. And in this case, using 1.6 micrometer beads, we could, before the curve started to go nonlinear, we measured north of 3 million particles per mil. And that's for all of our different kinds of consumables. And that's very encouraging. And the reason that happens is that the particular pores and membrane technology that we use here is such that the pores are very nicely defined and well spaced apart. So it's not like a fibrous membrane in which you're going to get strange capillary flow in which particles are going to stack against each other. In this case, because the pores are nicely defined and far apart, as soon as one particle blocks a pore, it will block that pore. And the fluid mechanics are such that the streamlines will be diverted to the next open pore, and therefore limiting the amount of stacking. So we can get high linearity, and that's proof of that. But now we've talked about black and white imaging and counting, if you will. What about particle identification, particularly in high throughput? The Aura enables fluorescence membrane microscopy, adding to this background and membrane imaging that I just showed you how it works. How does this work now? Well, one of the first challenges that I think we need to circumvent is that there's this miscommunication that morphology and the shape of particles and their light intensity can tell you, light scattering intensity can tell you what they are just by looking at pictures. Some companies and techniques are looking at machine learning to tell the nature of protein aggregation. But in this paper from the Japanese Pharmacopoeia, in which they helped validate and understand the US National Institute of Standards and Technology, ETFE, subvisible particles, NIST made subvisible particles made of protein, made of plastic, to mimic the shape and the refractive index of protein aggregates. And for these three sets of images, two of them are proteins. And I'm going to give you 1 minute to think, sorry about that, 10 seconds to think about which of these are proteins, of these three sets of images. And that was less than 10 seconds. But if you guess these two, you have much better eyesight and intuition than I do. If anything, I would have thought that the top two sets of images look more similar, but these protein aggregates form more crystal-like structures. So at the end of the day, if you were to use a machine learning algorithm to train these particles to figure out, it would have given you the wrong results, right? So morphology and intensity, they're powerful, but they're simply not enough to do definitive particle identification, no matter the route. At the complete opposite end of the spectrum, no pun intended, is FTIR, or say Raman spectroscopy. And as a professional spectroscopist, you would only, these tools are incredibly low throughput, so they're only used at the point of failure in failure-based diagnostics. After several signal cleanups and limiting the libraries of choice, you might have your spectroscopist or yourself finding a single particle that has a 1550 per wave number peak. And you would say, hey, that's an Mi2 bond, therefore it's protein. But that's an incredibly frustrating process. It can be an exercise in confirmation bias because you're limiting all of your libraries of choice from the very beginning, and it just takes a lot of time, right? So if you really wanted to characterize a sample with millions of particles, I'm not exaggerating when you're closer to a human lifetime than to a routine measurement at that point. So how do we do this? How does Aura enable this? So it builds on BMI, but it now supports labeled fluorescence workflows using the most established dyes that have been worked on for the last several decades. Now, we use newer membrane plate technology for that. You may notice that these are now black plates. These black plates support labeled fluorescence workflows. And the first three steps are the same. You background the plate to characterize its background texture, you filter your sample, and then you measure it, right? So at this point, you have your sizing, your counting, your images, just like what we've been discussing before. But then at that point, you can add your dye of choice to interrogate the chemical nature of the particles present on that membrane. And then of those particles that have already been sized and characterized, we're going to measure their fluorescence intensity and measure positive fluorescent events, much like a flow cytometer would do. And we're going to use that to understand the chemical nature of these particles. Now, the images, how are they going to look like? How are they different? So to first degree, your human IgG aggregates, with these alpha blended fluorescent images, you can see that you can pick a color. In this case, they will look red. These plastic shards that are not protein aggregates, in this case, they've been colored blue. And blue here means that it doesn't fluoresce. And actually, it's our SIMI signal. And I mentioned this in passing, but we have a light source that illuminates particles at an angle. And essentially what that means is that particles that protrude out of plane scatter lots of light. So this tells me, hey, this doesn't fluoresce, and it's actually sticking at a plane. And we can see images of a mixture of these two. Now let's take a look at this a little bit more quantitatively. In this slide, we have a scattergram in which in the x-axis, sorry for not having the label here, we have size. And in the y-axis, we have average fluorescent intensity. And each one of these dots is a particle that was measured. Now, this is the average fluorescence intensity using for the protein channel. And in this case, we use thioflavin T, which is one of the most well-established protein aggregate dyes that has been worked on for the last 40 plus years in Alzheimer's research. We can see that we get two different populations, right? So the higher you go in this y-axis, the more each particle fluoresces. So we have a population of particles at the bottom that do not fluoresce, while we now have a cloud of particles that fluoresce above the background. Because the system is powered with an expression engine that enables you to crunch all of the data for every single particle in context of the whole experiment, we could actually see that this fake protein aggregate made out of plastic, only it was called the protein 1.5% of the time, so very low false positive rates, while positive protein events were identified pretty much all of the time using well-established label fluorescence techniques. Now, this is just one example of how do you distinguish between protein and plastic. One of the more common questions in biologics, particularly in AAV and maybe in monoclonal antibody formulation, is degraded polysorbate. So the most popular excipient used to stabilize biologics has actually been found to degrade in the fridge and to form subvisible particles itself. And the reason for this is that it oxidizes and it forms these degradation components that can result in large particles. Now in this case, we see this fluorescence image of a protein sample containing polysorbate particles. We can look at the independent fluorescence signatures for the fatty acids as well as the protein signal by using two different well-established dyes. Bo-Dippy, one of the best established dyes at looking at lipids, and THT, one of the best established fluorescent dyes for looking at protein aggregates. And we can see how if we plot the lipid fluorescence channel staining versus the protein fluorescence channel staining, we can easily discern these two populations. This particular sample had lots of degraded polysorbate particles, potentially more protein particles, and some particles that are potentially mixed between these two. Depending on the level of mixing and polysorbate degradation, these curves will overlap more. Now, this system is actually quite sensitive with a very low limit of detection south of 10 micromolar of lauric acid equivalents. Now, what this means is that it's competitive with solution phase staining assays that are the most novel solution phase staining assays present in the literature, but with the added benefit that you can actually go back and look at these particles and confirm in fact that they were there and that they responded to a protein dye. And you can understand as well as their particle size distribution and how their presence actually impacts your overall particulate content. And this can be done very easily. And again, this would be very difficult to do morphologically because polysorbate particles, they can look just like protein aggregates as well. Now the aura is a platform, right? So the previous two examples that I gave was using thioflavin T to distinguish protein particles versus non-protein particles, using lipid stains to characterize polysorbate degradation products. But you can actually ask the question to interrogate the nature of all of your particles in one shot using more than one technique. Another approach is leveraging billions of years of evolution in enzymatic digestion. This top left well shows human IgG aggregates on a membrane. You can see there's these black particles. After we add trypsin on top of that membrane and remeasure that well, you can see that all of them went, all of them disappeared, showing that trypsin in fact cleaved and digested all of these protein aggregates. Here we have a mixture of polysorbate and plastic particles. When we used a cell lysis buffer, a lipid digestant, we actually removed these polysorbate components, and only the plastic particles remained. So this is visual, but it's also quantitative. And in one more example, in this case we have the same IgG aggregates that I showed you before. First we see the total IgG aggregate population measuring 35,000 particles per mil greater than 2. When we apply a rinse to those samples, we remove about 5,000 particles. This is an IgG wash sample, meaning that roughly 5,000 of the 35,000 particles were water soluble. Then to that sample, we trypsinize it. We apply trypsin. Again, all of this is done on the membrane, and we depleted about 90% of the sample, with only a few thousand particles per mil left. And again, the negative control is held. We possibly could have digested the sample more using more enzymes, but it basically proves the protein presence in the sample. Again, because trypsin has evolved for billions of years to digest protein specifically. Now, how is all of this information used to characterize AAV? Well, most importantly, AAV is very difficult to produce. A lot of the challenges in the field of AAV are related to scale-up, so low volume analysis is essential. Having a good understanding of your product is key from the earliest of stages. But that was very difficult to do in the particle context, in the subvisible particle context, because the existing tools just require too much material. Now let's see what we can learn with just 10 microliters of material. We have here 3 different sets of serotypes, AAVs 2, 5, and 8. The top 3 images are for unstressed sample, while the bottom 3 images are for stress sample at 73 C, so quite a bit of temperature for 2 hours. So what are we going to learn? So first we can see that for these membrane images, it doesn't look like AAV2 produced many particles without under no stress. AAV5 already showed the presence of some visible particle formation, while AAV8 produced large particles, even without stress. Again, but these samples haven't been stabilized. Now, when this same sample was stressed for 73 for 2 hours, we can see that they have very different types of personalities. AAV2 forming these very large discrete particles, AAV5 forming these fiber-like particles, while AAV8 showed more of a smearing, kind of a denaturing type effect. So that's quite interesting. Now let's get some more information. First, let's confirm that in fact we're looking at AAV aggregates. When we apply a protein stain, essentially all of these particles lit up, indicating that it's protein positive events. The blue channel is actually the height channel, if you will, the side illumination membrane imaging that I discussed before that allow us to characterize if particles are protruding out of plane. Now, In the case of unstressed, there's no response. The thermal stress, there's quite a few of these AAV aggregates. The rotated stress is quite interesting. We see a few particles, but a lot of them are blue, showing that they're tight and compact and protruding out of plane. And we see a similar phenomena for freeze-thaw. So far less particles than in thermal stress, but still present and actually quite big. And of course, our negative controls hold. But this can be done to characterize formulations and different serotypes very quickly under different conditions. Here's the characterization for these 3 different serotypes without and after thermal stress. But if we focus on AAV2, this chart on the right, we have the 4 stress conditions, so no stress, this thermal stress, which clearly produced lots of aggregates, north of 500,000 particles per mil, and the rotation and freeze-thaw stress. So both rotation and freeze-thaw stress produce quite tight data, so tight CVs, and they produce a little bit more particles in the control, with freeze-thaw producing every bit so slightly more. Now, this is all particle stability data looking at the fact that these AAV particles produce micrometer scale particles because they're unstable. Now, oftentimes, people have resorted to techniques that require lots of volume and therefore foregone subvisible particle analysis of gene therapies altogether. And maybe you might think, hey, I might not need subvisible particle analysis. I already have access to TM data. There's a great paper that came out about a year ago in PharmaRes that characterizes the TM for these 3 different serotypes. In this case, AAV2 had a TM of 67C, AAV5 had a melting temperature at 89, while AAV8 had a melting temperature at 73C. Now, as you might remember, we actually stressed these samples at 73C, but let's see what we learn. In the case of TM, you would imagine that AAV2 has the weakest stability, right? So at 73 Celsius, you will have passed this melting temperature. You're going to get lots of unfolding. But actually, when we looked at the subvisible particle stability, AAV2 was the one that produced the least particles. So my main comment here, observation, and many of you know this, is that melting temperature does not predict particle formation. It's valuable, but these are completely orthogonal effects. And even in the best of cases, many of these samples without stress wouldn't have passed the USP 788 requirements for the tolerable amounts of particles present in a sample. Now, the good news is now you don't have to choose because you can actually quantify and qualify to the single particle level and single particle identification level for these AAV aggregates. Now let's look at some more applications of this technology. So quickly, we're going to look at cell line development, in this case for monoclonal antibody production. We do have applications for other spaces as well. So as many of you probably have, lots of groups have very well established ELISAs to quantify titer and beyond. We, as a platform, the Aura enables one to use your existing fluorescence workflow of choice, whether it's a sandwich ELISA type approach, a direct antibody labeling with Alexa 488. You can do that. And in this case, we have a mixture of human IgG particles, subvisible particles, with lysozyme subvisible particles. And we can see how we can distinguish between different kinds of proteins, antibodies, and lysozyme crystals using antibody labeling without having the need to potentially get confused with direct morphological analysis. One way this can be applied in the field is we can determine if secreting cell lines are producing aggregated product from the ground up. Now, I understand that in cell line development, what is king is the ability to produce titer, right? That's what it all comes down to. It is manufacturing at the end of the day. However, you could be producing junk from the ground up. And what you can see here is that this particular cell line secreted antibodies, sure, but they were all aggregated. And this was measured using a specific antibody against that product. And one advantage that this system has over other technologies when it looks at characterizing these types of samples is that you really can't break it. You can't clog a membrane. So if you wanted to take a look at this in the context of unpurified, unfiltered solutions, even in the presence of CHO cells and whatnot, you could. And you could still get some very valuable insights about what's happening. Now, many of you may question, hey, are you actually measuring the product aggregating, or is it just the fact that you use the wrong antibodies or your antibody labeling process is unstable? And the short answer is no, is that we basically get no signal from the primary and the primary but secondary antibody by itself. We're actually looking at aggregated proteins that were produced from these cell lines. And because the Aura is a platform, this might be a little bit of a busy slide, but it can adapt to your workflow. In this case, we've done a lot of work with solution phase staining. That's a more traditional way to do surface chemistry, right, where you incubate samples in solution. You could also dye or apply dyes or enzymes on the membrane directly. And each of those can be optimized very quickly because, again, it only takes 1 minute per sample. So one great advantage of this technology is that it doesn't put you you're back against a wall. You can really optimize your assays very quickly and get valuable particle stability and sterility and contamination information quite quickly. Now, many of you may be here to see how can this technology help me characterize my cells, and particularly in the case of cell therapy. And it's a stability, viability, and impurity platform to get vital answers, as early or as late as necessary. But subvisible particle analysis has actually been quite challenging in cell therapies. And the reason for that is that cells can also be subvisible particles, right? Some cells can be they're all north of a micron, and it's very easy for them to be larger than 10 micrometers and therefore not pass USP 788. On top of that, the current tools are basically not designed to handle cell therapies. Cell therapies can have over 1 billion cells per dose. Cytometers are also not particle sizers. And then the legacy subvisible particle analysis tools like flow imaging and light obscuration, it's already been published that they miss up to 50% of the cell samples that are being measured. And the reason for this is that you have like a tall microfluidic channel, call it 300 microns in size, and it's only focused from an optics working distance standpoint. You're only focusing on about 20 micrometers of that flow cell. So magically, things are flowing above and below the sample, missing critical information. On top of that, these fluidic-based systems, they tend to clog, presenting a limitation of working with high concentrations, and they're very low throughput, and they're easy to break. So cell therapy subvisible particle differentiation in the AuraCL is easy. And in this case, this is a typical plot you would see in flow cytometry. This is solid phase cytometry. And in the y-axis, we're plotting DNA staining, and in the fluorescence for every particle. And in the x-axis, we have the average protein staining signal for every particle. So this is quite interesting. We can see that the particles that are responsive to protein staining are protein aggregates, potentially formed from, say, the HSA that is used to stabilize many of these cell therapies. The cells respond to both cell and nucleic acid signals, while non-biological particles stain, and potentially some more DNA aggregates, they respond to DNA staining alone. What the system can help you characterize is the presence of doublets, triplets, quadruplets, or even large cellular aggregates present in the solution. And again, these large aggregates, they could potentially cause capillary occlusion and can be even visible in nature. And the reason why they fluoresce with a nucleic acid stain is because they're actually cells. Now, this system allows you to analyze visible particles. That means particles larger than 100 micrometers in size, and not only measure them, but actually distinguish them and understand what they actually are. And this, you may remember this image that I showed at the beginning of this presentation, but it was actually a fiber embedded in a cell therapy product. And you can quickly determine that without having to recur to a complex Raman system. Large aggregates, in the case of gene therapy, like a very large visible aggregate, It fluoresced in thioflavin T, indicating that it is proteinaceous in nature, and therefore it is the AAV. What are some more key insights that we can get? Now remember, this technology is high contrast, so it's very sensitive to picking up particles that could be easily missed with fluidic-based techniques, including light obscuration, flow cytometry, or flow imaging. Here we have a serial dilution of a concentrated cell therapy, and what you can see in the black line, that's the total particle count population, but the red line is actually the population that responded to a nucleic acid stain. So this is my cell population. You can see here that there is an obvious gap. So about 25% of the sample here was not cells. So there's something else that happened with this particular cell therapy. Now let's dig a little bit more into that. But first, one key thing that this technology can help you characterize is actually cell viability. In this case, we're using very well-established cell viability techniques to distinguish between live and dead cells. We're looking at a monocyte population, and you can see that typical monocytes, they tend to be larger, but when they're dead, they tend to be hyper-compressed, as these arrows point to here. And using a very well-established viability assay, the first thing that we do, let's focus on this image on the left. We use a very low concentration of a DAPI stain, which is a nucleic acid stain. So at low concentrations, this nucleic acid stain will only permeate through dead cells because those walls have been compromised, and therefore those light up quite strongly. However, when we remeasure this exact same sample, right, and that's a key advantage, we're not measuring a different sample, but the exact same sample on that same membrane, it's now subjected to a high DAPI concentration, this forcing gradient will make all cells fluoresce. And we can use these differential staining properties to understand all cells versus dead cells. Now, if you see here, we corroborated this information across different cell types and across different platforms. In the case for both T cells and monocytes, the results obtained for cell viability for the aura match both flow cytometry and hemocytometry. In the case of aura, we use DAPI. In the case of flow cytometry, we use propydium iodide. And in the case of the hemocytometry cell counting chamber assays, we use a trypan blue cell obscuration assays. We can see that all of these results are very well within error and comparable to the most established viability techniques. But I understand that you've already have some of those. One key piece of information that you can get with these high contrast, high sensitivity technique in the case of aura is that you can characterize viability in the context of product purity. Now let me walk you through this example. And we have a cell therapy sample that came from a fresh culture compared to one that has been subjected to several rounds of freeze thaw. Now in the case of the untreated sample, first we see that 86.1% of the sample was actually cellular, while 14% of the sample was non-cellular. And we'll take a look at that in a minute. But if we focus on the cellular sample, of those that were responded to the DAPI staining, 82% were live cells, and about 19%, 18% were dead cells. So, and of the remaining material, much of it did not respond to a protein stain, only 1.3%. Now let's take a look at the sample that has been subjected to several rounds of freeze thaw. About 80% of the sample detected was cellular and 20% was something else. Of the cell sample, only about 10% of these cells were live and 90% of them were dead. But of the remaining sample, this 20% material, most of it fluoresced under thioflavin T, under this protein staining dye, indicating that either the human serum albumin formed aggregates as a result of the freeze thawing procedure, or potentially there might be some cell debris that formed as the cells were dying. And we can not only get this information, but actually image it. So the non-cellular particles, in this case this blue channel, much of them protruded out of plane, indicating potentially plastic contamination. The protein and cellular debris were properly characterized, And the viable and the non-viable cells were characterized again using this differential staining assay that I described before. One last example is finding the needle in a haystack particle. This technology can be used to find leftover dynabeads in your cell therapy products. And what you're seeing here is a mixture of T cells and dynabeads. Now, the dynabeads are these magnetic particles. They come in different sizes, typically about 4.75 microns in diameter. And they're coated with CD3, which signals the T cells to activate and expand. In the aura CL, we can find these remnant magnetic beads that were not removed via magnetic isolation in the presence of these T cells and actually image and quantify them. So you can see these blue particles are your dynabeads. These black particles are some remnants that are non-cellular, while these red particles, they respond to a protein and signal indicating that they're cellular. Like I indicated before, we can produce these scattergrams. And here we can see our cell population. So we have lots of particles that fluoresce quite strongly with a DNA stain. We find this particle population hovering at the 5 micron line, likely coming from the dynabead particular product, and then this other cell debris or non-cellular material. And we can quickly get and characterize all of these materials using the built-in software technology that comes with the Aura systems and the particle view software. Now, one interesting light source that I mentioned before is this side illumination source. We can illuminate these particles at an angle, and what we can actually see is that the dynabeads, they're covering around 5 microns in size, which is exactly what we expect, but they tend to scatter more side light than the other materials, which potentially are a little bit more compliant. And that's really interesting because this hard material, centered around 5 microns, can be quickly discerned. And if you click on these, in every single one of these dots, you can go back and assess the raw images and confirm instantly that they are dynabeads, in fact. So why Aura? Honestly, why not Aura? It is the high throughput subvisible particle analysis for all biologics, whether they're protein biologics, gene therapies, or cell therapies. It's rapid and intuitive categorization of product aggregates and external impurities. It's compatible with USB 788 and embedded in chapter 1788, which allows for digital imaging. It enables low sample consumption so you can characterize the stability of your product as early as needed in the process. And it offers detailed information for particles across the entire size range above a micrometer. It's compatible with most fluorescence, protein, and cellular assays, whether you're using dyes or digestive enzymes. It's compatible with CFR. It's very easy to use. And honestly, I can't wait until you get your hands on one. Thank you so much for attending my talk. And I'm now looking forward to your questions. Thank you. So feel free to reach out to me, or for more information, please email us at sales@halolabs.com. Thanks so much again for attending. Bye.
Bernardo, wonderful presentation, and thanks for sharing all that data with us. We really appreciate it. I think the audience now has a really great sense of the RCL's power and capabilities and how it can be beneficial to their work. So thanks again. To the audience members, the Q&A is coming up in just a moment. So if you have some questions for Bernardo, don't be shy, send them on in. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. All right, since we have a bunch of really great questions lined up, let's move on to the Q&A session. Bear with us for just a moment as we make the transition into that Q&A session.
All right, everyone. Thanks for joining us for the Q&A session. We appreciate it. Bernardo, again, great presentation. And we have a bunch of questions here from our audience, so let's get to them. The first question, one of our audience members would like to know, what formulation should we use to avoid protein AAV and cell aggregation? Is there any specific surfactant or detergent that we can use to avoid these types of aggregation?
Hello, everyone, and thanks for the question, Jeff, and the audience. Unfortunately, there isn't a magic bullet for a single surfactant that you can use to avoid protein, gene therapy, or cell therapy aggregation. And actually, if that was the case, you wouldn't have to do pretty much any formulation development. Every biologic has its own stability profile, meaning that it's going to need different types of surfactants and excipients to keep it stable and to keep it from aggregating. The things that formulators need to consider are using a range of pHs to keep their biologic stable, a range of salinities to keep their biologic stable. On your surfactant wheelhouse, there are different surfactants. The most popular ones are polysorbate 20 and polysorbate 80. There's other excipients like sucrose, and so it's formulation development is a whole world to its own, but it's really one of the most important steps that turns a biologic candidate into an actual product. This tool allows you to quickly identify the formulation conditions that are best. So the combinations of these pHs and excipients and surfactants that allow you to produce the best product with the least aggregates, whether these are proteinaceous, gene therapy products, or cell therapy products.
All right, thank you for that, Bernardo. Next question, one of our audience members would like to know is, do you use a protein-based standard for calibration purposes?
So because we measure particles, right, and in the case of protein aggregates, so visible protein aggregates, or say even gene therapy AAV aggregates, there's a challenge, right, because the particles that can form are highly heterogeneous, right? So actually, the U.S. NIST came up with a protein mimic particle called ethylene tetrafluoroethylene, which basically mimics the size, shape, and morphology and particle size distribution of common protein aggregation profiles. So to calibrate our systems and particularly show the ability to measure polydispersed samples, we use NIST ETFE.
All right. Next question an audience member has is, are you limited on the dyes you use, or can I do other things? For example, RNA-specific labels like cyber gold or ribo green?
That's a good question. No, so the Aura is a platform, and it's very flexible, and you can use it's pretty much any dye of choice that you're currently using in your workflow. Using dyes like Alexa 488. You can look at nucleic acids or DNA stains like the ones that you measure, cyber gold. So no, you're not limited. There's a couple of default stains that we recommend that go out with the system, like thioflavin T for protein identification, DAPI or host staining for cell staining. But in general, it does the system is flexible enough to adapt to pretty much any dye of choice.
All right, great. How do you deal with mixtures such as residual host cell proteins? Can you label specific proteins like an ELISA type assay?
Yeah, so that's really interesting. So one of the way that we deal with mixtures is to use product-specific dyes. So just like you mentioned, if we use ELISA-like assays like antibody labeling, we can specifically label the drug product or the target protein of interest while not staining the rest of the protein content. So one way that we do this is via using antibody labeling, and we have a lot of applications, whether it's for specific protein identification or even in cell line development, right, to show these developers that oftentimes the proteins that are secreted from the cells are actually aggregating, right, from T equals 0. So antibody labeling is a very popular technique with this platform.
All right. And next question the audience member has is, how much time does the fluorescence measurement add?
It only adds a few seconds, probably about 5 seconds compared to the bright field, because they can be acquired at the same time. So very minimal time. It doesn't really double the measurement time. It only adds um, a few it only adds a few seconds to the measurement.
All right, great. And what concentration of dye is needed to sufficiently label the particles?
So that's a good question. It's protein and application specific, but anywhere from very low concentrations of down to 5 micromolar of, say, the Bo-Dippy stain for polysorbate identification, to, if you have a very soluble stain like thioflavin T, you can use almost millimolar concentration of dye. So it is a assay dependent question, but from very low to quite high concentrations. And one of the good news is that assay development is quite fast in this system, right? Because it's high throughput. So if you want to hone in on, for example, a particular dye concentration, you want to use as much dye as you need to get a strong signal, but you also don't want a high concentration of dye to start resulting in precipitates. This system can help you very quickly hone in on that perfect dye concentration to get the most signals and the least amount of dye precipitate, so that you don't to get the best signal and you don't impact your measurements.
All right, great. Our next question, an audience member would like to know is it possible to distinguish between different AAV serotypes with this technology?
We can distinguish between, so that's a good question. So so there are antibodies that are designed to label different kinds of serotypes, so adapting those types of methodologies. We could use it to distinguish them. But again, this technology at its heart is a physical stability technique, right? So you could distinguish between different kinds of serotype aggregates as opposed to different serotype monomers. So it would help you distinguish whether if you have a mixture of gene therapies, for example, of serotypes for a gene therapy product, you could determine if one particular serotype is more likely to aggregate than another, as opposed to tell you, the percentages of the individual monomer concentration of AAV.
All right, great. Thanks for that. Next question, interesting one. Do you have any data that shows aggregation levels over time?
Yeah, and I'd be happy to share that with the audience member, and absolutely. And it's a great question, and the aggregation versus time dependence is something that's very important to account, right? There's a lot of variables there. So, for example, environmental conditions. Are we talking about aggregation on a refrigerated sample, and then on which sample are we talking about? Is it a gene therapy, a protein product, or a cell therapy? What excipient and formulation conditions are used? So all of these matter. Oftentimes, we tend to think that over time things are much more likely to aggregate, but in the case of, for example, polysorbate aggregation, oftentimes heating or these types of thermodynamic effects of just leaving samples laying on a surface can actually make them dissolve and fall into solution. So the short answer is yes, and I'd be happy to share it.
All right. Well, thank you for that, Bernardo. Next question, important question, I think. How is the membrane compatibility with various excipients, for example, high concentrations of DMSO?
So the membrane is compatible with DMSO. Typically, we use up to 5% DMSO on the membrane, which, even though it sounds low, it's compatible with most, for example, small molecule solubility experiments. So it can take up to 5% DMSO, which is quite high concentration, and below. And it is compatible with a range of organic solvents up to about the 5% line.
All right. And one audience member would like to know, how does the sampling efficiency of AURA compare to MFI and FlowCam?
It's much higher. So AURA, as everybody saw, so we sample 100% of the sample because all of the particles are in focus, and 100% of the sample goes through the membrane. In the case of the flow imaging techniques, they're flown in microfluidic channels, which are very tall, right? They're about 300 microns tall, but the depth of focus is only about 20 to 30 microns. So it's only a fraction of the sample that is truly in focus, right? So sure, does light hit all of the sample as the particles flow through? Then sure, but only a fraction of that light is in focus, and that's one of the reasons why for these new cell therapy products they found that the sampling efficiency is actually on the below the 50% line, right? And it's because the focus is in just such a small swath of where the entire sample is falling through. Well, in our case, there's not that problem because 100% of the samples are imaged on the same plane.
Gotcha. Makes sense. Next question. How much flexibility do you have in fluorescence workflow? Do I have to stain on the membrane or in a solution?
We have lots of flexibility. The Aura is a platform. So you can do solution phase staining or membrane phase staining. Oftentimes, many of our users like the membrane phase staining approach, in which first you filter and measure your samples, and then you ask the question, hey, of these particles that I image size counted, and I got particle size distributions, which of those are proteins, cells, polysorbate, right? So you can ask that question after the fact. One nice reason to do that is that you're 100% sure that your dye is not perturbing the physical stability of your sample, right? And that's why it's a preferred. It is a good way to go about things, but the solution phase staining is much older and established. So, for example, with a lot of our nucleic acid stains, we perform them in solution because we only use a very tiny amount of nucleic acid, right? For example, when doing these viability measurements. And the good news is that they're both compatible. And if you have any questions, we'd be happy to walk you through it. But ultimately, because the tool is fast, right, you can hone in on your preferred method of choice, understanding that both of those have, as with anything in sciences, their pros and cons.
All right. And Bernardo, how do you ensure fluorescent dye particles are not confused with stain particles?
And it's back to my previous point in that if you first measure the particles prior to introducing dye, so that essentially you're making sure that if you use your first workflow, which is just measure particles without adding dye, the software understands where your particles live on the membrane. Then if you apply a dye after the fact, there's only a small region of the membrane, right, that is populated with these particles that you've already measured. So if there's, for example, precipitate in the dye, it's very unlikely that the precipitate falls on top of the particle that you already measured. So by doing membrane phase staining, it is one of the reasons why a lot of our users like that approach, in that it accounts for any potential dye precipitate type undesired dye precipitate effects. So membrane phase staining is how we would recommend mitigating that problem.
All right. And next question, I think, is a good one for all of our biopharma and biotech audience members out there. Audience member would like to know, is the method compatible with USP, and can it be validated?
Yes, this method is compatible with USP 788. Digital membrane microscopy is in Chapter 1788, and therefore there's a direct pathway to validation.
Excellent. Looks like we have time for two more questions here. One audience member would like to know, what's worse, fewer large particles or a lot of small ones?
I think fewer large particles, because they can potentially, depending what we mean by large, but certainly above, say, 50 microns, very large particles can cause capillary occlusion. And those are really the ones that we're trying to mitigate with also visible particle analysis techniques.
All right. And our last question is, what is the size limit on the higher end?
5 millimeters, one whole well worth of diameter. So we can easily and routinely measure fibers as long as 5 millimeters in size. And of course, particles as small as a bit larger than a micron in size.
Right, excellent. Well, thank you, Bernardo. Appreciate that. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or you can feel free to forward the link to any of your friends and colleagues, which we always recommend. I'd like to thank Bernardo again for his very informative presentation, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Halo Labs for sponsoring this webinar. Hopefully, we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy and get your vaccines.